All right, everyone, it is my pleasure to introduce our next speaker. Maia Jones is a third-year PhD student at Washington University in St. Louis. She's studying with Dr. Castanea Edwards and Dr. Matthew Albrecht of the Missouri Botanical Garden. And Matthew is on our board for the Ozark Chinquapin Foundation, and he's been very instrumental in making things happen over the years. And we've had a number of cooperative projects with the botanical gardens, and we've benefited as an organization from that cooperation and generosity. Right now, Maia is working on her thesis, which focuses on applying population genomics to design ex situ collections of Ozark Chinquapins and Alabama snowreed. She earned her bachelor's in organismal biology from Scripps College and previously worked as a research assistant at the California Academy of Sciences. She studied the conservation genomics of endangered cycads. Maia's research focuses on using population genetics, quantitative genetics, molecular systematics, and DNA barcoding to help understand the ecology and evolutionary biology of endangered species and to aid in applied in situ and ex situ conservation efforts. And if any of those words don't make a lot of sense to you, I am confident she is going to be able to demystify those. So if you would, let's give her a big round of applause and welcome her to the stage.
Hi everyone. Can you hear me okay, or should I?
If you want to take that out and hold it, that would be better.
Okay, is this better?
Yes.
Okay. Thank you for the introduction, AJ. Yeah, I'm just really excited to be here and be learning from you all. I'm pretty new to studying Ozark Chinquapin, so it's great hearing from people who have so many decades of experience working with this species. And thank you, Steve, for having us come from the Missouri Botanical Garden to share what we're working on. Yeah, so as AJ said, I'm a third-year PhD student at Washington University in St. Louis, and I'm doing the research portion of my degree at the Missouri Botanical Garden, and I'm part of this larger project that's funded by the Institute of Museum and Library Services, and we are, I guess I can go to the next slide. So overall, the project is understanding and improving the conservation of interest-specific variation of imperiled plants in ex situ collections. So essentially that is capturing the diversity of wild populations in botanical garden collections. So as we know, we are seeing global declines in biodiversity. Over 40% of plant species are threatened, and most of these threats are anthropogenic drivers, so including habitat destruction, land use change, introduction of invasive species, and climate change. And so some of the ways that we can mitigate the negative effects of these threats is by intentionally safeguarding biodiversity. And so there are two primary methods that we can use. One is in situ conservation and one is ex situ, and I'll get more into these and explain what they are. So in situ conservation is conservation of wild populations in their native habitat, and this is our preferred method, so I think this is what most of you all are familiar with. And it usually involves active management of the habitat using prescribed burns and removing invasives. And then ex situ conservation is conservation outside of the natural habitat. And so this consists of creating seed banks or collections of whole living plants. And in particular, the living collections are useful for species that we would call exceptional, and that means that they have seeds that cannot be banked using traditional methods such as freezing or drying. And these ex situ collections are designed to represent all the diversity that we see in the remaining wild populations. And so these seeds or living plants are maintained to produce material for reintroducing into habitat or augmenting those wild populations or for restoring the habitat overall. And so we kind of want to use these ex situ collections to supplement what we were doing with in situ conservation efforts. But there are some challenges with designing ex situ collections, and so we want them to be effective, meaning that they are conserving the wild diversity that's present, and so ideally that would mean having seed from every individual so that each individual is represented. But then knowing that the space and resources that we have in botanical gardens is limited, we need to be really efficient about how we're capturing all of this diversity. And so there's a little bit of a trade-off there. And so the standard approach for building these ex situ collections comes from some general collection guidelines that were written by folks at the Center for Plant Conservation. And these are methods that are focused on sampling seeds in a sustainable way so that we don't deplete the seed crop of the wild populations. And some studies that have evaluated the utility of this for several threatened species found that this doesn't necessarily work for all species because you're trying to sample in a representative way, but you don't actually know the genetics of the plants that you're sampling from, and so you don't really know what kind of diversity you're collecting. So that's where this alternative approach comes into play, and so this approach involves assessing neutral genetic diversity, which is variation that's present in the genome of an individual, and this variation arose through random processes, so it's not under selection. And then we also want to evaluate population structure, and this helps us differentiate between individuals and between populations. So I know this plot is kind of a lot, so I'll explain what's going on. So this is from a different project that my advisor, Christine Edwards, did with her lab on the Missouri bladder pod. And so they sampled from populations from throughout the range and genotyped them. And then, so essentially, this plot at the bottom with the bars is called the structure plot, and each chunk is a different population, and I know you won't be able to see from the back, but each of those chunks is made up of bars, and each bar represents one individual, and so they're color-coded by the proportion of a certain type of ancestry they have. And so for this particular data set, there are 8 different, let's say, sources of ancestry, and then in this analysis, we can attribute a certain proportion of that ancestral group to each individual. And so if an individual is completely in like a solid color, then that means that that individual or that population has become more genetically isolated from other individuals that are made up of different colors. But then for bars that are mixed, that means that there has been some evidence of shared genetic material in the past. And so what we can get from this is kind of an idea of how the populations are related to each other, maybe how genetically isolated they are, and this is really important for understanding what kind of genetic diversity we have in our collections. But besides neutral genetic variation, there are other types of variation that we want to take into account. So there is adaptive genetic variation, which refers to regions of the genome that are under selection in response to some change in the environment. And then we also have neutral phenotypic variation and adaptive phenotypic variation, and phenotype is the observable morphology of an individual that we can measure. So you see in these plots, I guess I can use a pointer. So for neutral genes and neutral traits, the value doesn't change even when there's variation in the environment. So for example, this map is showing the average monthly temperature of April, I believe. And so you can imagine that a plant growing here is experiencing an environment that's very different from the plant growing here. And so those plants are responding to the changes in the environment, and so that's where the adaptive genes and adaptive traits come in. So we see that those values vary as the environment, some given environmental variable is changing. Sorry. But why do we care about including both neutral and adaptive variation? So adaptive variation is often found in individuals that may have acquired some kind of advantageous traits for survival and reproduction in their local environment. And so we would call that adaptiveness. And then neutral variation found in the genome could contain the potential for adapting to future changes in the environment, and so we would call that adaptability. And so, like I mentioned before, we have limited resources when we're building these conservation collections, and so we have to find a trade-off between including individuals that are adapted and adaptable. Because if we were to say like, oh, well, these individuals are adapted, we want to have all of those, that's narrowing down the gene pool, and it's limiting the amount of neutral variation that might make an individual more resilient to future changes in the environment. So it's really important to have a combination of both, and we have to kind of calculate what that trade-off is. And then I also wanted to highlight why we are looking at phenotypic variation, not just genetic variation. So yeah, phenotypic variation is the observable morphological variation, and as well as the timing of life history events. So this could be the size of leaves or the number of leaves. Of course, when the plant gets too big, you can't count every single leaf. But we're also measuring stem width and tracking germination time. And so for this project, we are growing plants from seed in a common garden setting. So if we have them all growing in the same environment and getting the same treatment, any observed phenotypic variation that we see could be due to adaptation in the environment that those seeds originally came from and that were inherited. And so we're looking at phenotypic diversity because it could indicate some adaptive potential for reintroductions. And then it's also important to note that these different forms of genetic variation can be distributed differently across the landscape. And so if we look at the top of this map, oh, kind of cut off the bottom, but this is an example from a species that is an herb in the Amazonian savanna. And so in the map, each dot is one individual. And then, similar to the structure plot that I showed, they are color-coded by assignment to a genetic cluster. And so the top one is showing the neutral clusters, and if we look, we see there are 1, 2, 3 clusters. But then if we look at the adaptive data, there are 6 clusters. And so it's really important to be able to tease out the neutral and adaptive variation, because if we were only looking at the neutral variation, we could be missing out on all of this variation that we want to have included in our collections. And then, so bringing it back to the Ozark Chinquapin, we can see that there's a lot of environmental variation across the species range. So this is a BONAP map from 2014 showing the range of Castanea ozarkensis, and this is a map of monthly averages in February. So you can see like these populations in southern Missouri are experiencing a different February climate than maybe the ones in northern Louisiana. And so they are probably experiencing different summer highs and winter lows and precipitation levels and the timing of frost and different soil types. And so all of this environmental variation could be leading to selection on different genetic or phenotypic traits, which could lead to local adaptation. And it's important for us to understand whether or not there is local adaptation, because I know for a lot of the work that y'all do, you're reintroducing seeds or seedlings out into habitat, and if we want to ensure a higher chance of survival, we want these seedlings, we want to have knowledge of the genotype of these seedlings, and we want to reintroduce them into habitats that they are locally adapted to. Otherwise, it could have negative effects on their chance of survival and future reproduction. And so we want to understand how this neutral and adaptive diversity is distributed across the space or across the landscape, so that we can even target specific parts of the species range for protection and for management. So some study questions for our project overall. Do we see evidence of local adaptation in imperiled plants? If so, are the neutral and adaptive variation in both phenotypes and genotypes structured differently across different spatial scales? And then how do we balance trade-offs in representing neutral and adaptive variation in both phenotypes and genotypes when we only have a finite amount of resources for managing these collections? And so in this project, we have 3 herbaceous study species, an aster, a bladder pod, and a prairie clover, and these are the species that we can seed bank. And then we have these 2 woody species, and so these are the 2 that I'm studying for my thesis. The Alabama snowwreath, it does produce flowers that are pretty showy, but it's never been seen to set seed. And then, I'm sure as you all know, the Ozark Chinquapin, you can't freeze the seeds and then expect to have them germinate, and so we can't use traditional seed banking methods for these 2 species, and that's why we're building living collections to manage these species. And so that's why being at a botanical garden is so helpful for this kind of project, because we can care for these living collections, and then we also have growth facilities that we can use for our common garden experiments, and then we also have a conservation genetics lab where we're doing all of our genetics work. So the current phase that we're in is collecting both the phenotypic and the genetic data, and then down the road we plan to do associations with the environment, so between genotype environment and phenotype environment, to kind of figure out like which traits are neutral and which are adaptive. And then at the end of that, we're going to synthesize it into kind of figuring out what the trade-offs are in building this collection and incorporating different types of variation within the species. Okay, so focusing in on the Ozark Chinquapin, one of the main questions that we have for this project is what are the species limits and conservation status of the Ozark Chinquapin? And so we can see that it has range overlap with the much more common Allegheny Chinquapin, and this has implications for the conservation status, because as it is currently, the Ozark Chinquapin has a pretty limited range, and so it's of more conservation concern than Castanea, which is, I believe, its least concern. And so if we take one trait in particular, such as seed size, we know that there's so much variation within Ozark Chinquapin seed size, and that it might have some overlap with the seed size of Castanea pumila, and that could make it difficult to kind of delimit the boundaries between the 2 species. And seed size is important because it has implications for like nutrient storage and energy storage when the plant is trying to germinate. And so another thing we want to look at are the genomic limits of the species. And so this is from a study from 4 years ago, and they were looking at Castanea ozarkensis and pumila, and so each dot is population, and this is similar to the structure plot that I was showing before. So again, each bar is one individual, and each color represents a different genetic cluster. And so we can see that in these areas where ozarkensis and pumila are either co-occurring or the populations abut each other, there is some evidence of past gene flow between the species, and that could make it more difficult for us to delimit the two species. So that is one of our main questions for the species. Are Castanea ozarkensis and pumila genetically and phenotypically distinct? And then once we can define what Castanea ozarkensis is, do we see evidence of local adaptation? And are there trade-offs in conserving both neutral and adaptive variation? And so we received over 600 seeds representing 54 maternal lines, and some of those are probably maternal lines are probably Castanea pumila. And I know that you all had a hand in collecting those seeds, and so I just wanted to thank you all for contributing those to our project, because we really could not have done this without all of your contributions. And so we grew those seeds in our greenhouse setting, and then collected leaf samples and extracted DNA. A lot of that is more detailed than you probably want, but if you have questions about it later, feel free to ask. So we basically are doing pretty in-depth whole genome sequencing so that we can look at all different regions of the genome. And we're using these data to analyze the genetic structure, to understand where the species boundaries are, and then we'll be able to use this data in order to conduct associations with different environmental variables and see which environmental variables are maybe involved in local adaptation. And so one of the main results is that Castanea ozarkensis and pumila do have a slightly different genome size. And this is something, I mean, maybe it's just interesting, but like on its own to some folks, but it also is important for the kind of reintroduction efforts, because if you think about having a larger genome size, it means that it takes longer for the cells to divide, and that means that it takes longer for the plant to develop, and the longer the life is, or the longer it takes to reach developmental stages, the less adaptable it might be to changes in the environment, especially as we know there are so many rapid environmental changes happening. And so that's just something that we want to be aware of. And then I also want to clarify, these are not our results, these are still from that paper from, well, it cut off the bottom, but from 4 years ago. We're still working on the data, and I didn't want to present something too preliminary and lead us astray. So I'm just going to talk about the kinds of analyses that we're going to do and kind of what we can take away from that. And so I'll go through them. So as I showed before, the population structure, this is used to estimate the ancestry of individuals and understand how the populations are related to each other. And then we also have what's called a principal component analysis. And so this allows us to condense all the genetic differences that we see into axes of variation that each explain a proportion of the overall variation. So we can see like this first axis, all this variation that we see along the x-axis is almost 7% of the overall variation that we see. And so in this plot from this previous paper, each color is a different genetic cluster, and we can see like which clusters or which populations may be more genetically related to one another. And then if we want to look on an even more granular level for the relationships between individuals and populations, we can use a phylogenetic tree. This shows evolutionary relationships and can be used to determine the limits of a species. Yeah, so kind of to reiterate, these analyses used together can help us understand the evolutionary history of the populations, determine if there was past gene flow, and define the species boundaries. And so, down the road, we are going to continue analyzing our data to clarify species boundaries, look for genetic diversity, and determine whether we see local adaptation in the Ozark Chinquapin. And then other things that we're interested in exploring as the plants get bigger, we want to measure some physiological traits and reproductive traits, and also do some blight resistance assays. And then once we are able to figure out what the trade-offs are in building these collections, then we can also make conservation recommendations, both for those ex situ and in situ conservation efforts. So overall goals for this part of the project are to resolve the limits and the species limits and the conservation status of Castanea ozarkensis, and get a better understanding of how this within-species variation is structured, both within and between populations across the landscape, and then hopefully share out what we've learned about conserving the species and how we can incorporate different types of variation. So yeah, I'd just like to thank our funding source, MLS again, and all of our collaborators, and once again thank Steve and Leslie Bost for having us here and giving me the opportunity to talk about the work that we're doing. And this is my email if you have questions. I don't know if we have time for questions now.
Probably.
Yeah, all right, thank you.
This time in Latin. We have something for you. Okay, so I went to Missouri Botanical Garden up there to talk about a great group of people and do cutting edge research and technology. And it's been helping us out a lot. And this study that y'all are doing is a total of, was it 6 different species altogether? It's what now? 5. It was 5, okay. And so they went ahead and bumped one species they were doing the study on, which was the, what now? Okay, that is the, common name on that.
Yeah, I can't remember which kind of oak it is, but.
Yeah, but anyhow, they bumped a species out to go ahead and do the study for us. So we were working on a grant of $2.3 million a few years ago through USDA, and we almost got it, believe it or not. And so we contacted Missouri Botanical Garden to find out how much this study would cost, to find out specifically, you know, when you say phenotype, you're talking about, I think like photo, what it looks like. Then genotype, you know, the genetics of it. And so this is real important stuff to help make sure when we do a breeding program that we are using the right components to breed for resistance. We don't have something in there that does not belong or not adapted. There's a lot of confusion between the familla. I was looking at this map right here, and we got to map some of that we've got verified American chestnut, Ozark Chinquapins. And I need to come up and spend some time with y'all and say, this sample right here is one that we know is a tree, and so there's a lot of confusion on it. But anyhow, so went up there, and so we had a Zoom meeting, and we talked about how much this study would be, and if I remember correctly, this particular study was going to run somewhere around $470-something thousand, I believe, wasn't it, Leslie, somewhere in that neighborhood, for Missouri Botanical Garden to do it. They're hiring new people to do this. So then I was contacted by Christy and also by Matthew Albrecht, and he said they're going to go ahead and y'all picked up the Ozark Chinquapin from one of those species. And so as a token of thanks, we have for you a thermos right here. And so you may have to keep this away from Z over there. She may try to wrestle it from you. As a token of thanks, we had it laser engraved and custom made, and there you go.